Welcome to this edition of Side Nerds Podcast. This is podcast number 2 for cell biology, and we will be looking at cell structures and functions. Isn't he cute? Minnie the dog. This is a short introductory video that I found on YouTube. And I thought it was a really cool way to introduce a unit, so take a minute or two to watch this carefully as it will unfold some of the things we will talk about in this unit. So, this is cell biology, and cell is all about the cell. You can't talk about any living thing without talking about the cell, because it's a very essential element. Basically, if you want to describe a big elephant, you have to describe the elephant on this very cellular level. And so this is our journey for this unit. As with all of our other units to come and the one previous, like I told you before, you will have a set of focus questions and look at it as a pre-study guide. So as you go through the unit, you should be able to answer each of these questions, and by the conclusion of this unit, you should be able to know the answers to these questions, because that's pretty much what these questions are what actually the unit is about. You should be able to describe different types of cells, be able to describe different what a cell should look like under the microscope. You should be able to tell, what the cell theory is and how those are put together. You should be able to talk about the different organelles in the cells and what to do, and being able to describe how things go in and out of the cells, and when does the cell use energy, when does it not use energy, and so forth. Here are some key vocabs for this unit. Like always, define these terms, and they'll be able to help you to understand these concepts and describe in this unit. Like any language, if you're going to speak that language, you got to learn the words that go over that language. You can't say you want to speak French if you don't know how to say a French word or two. So before we go any further into the word itself, this unit, and just to caution you, this unit will start out in the first 9 weeks and will enter into the second 9 weeks. So please don't be like, oh, we're already in the second 9 weeks, so this is first 9 weeks. Please follow along, please. But before we go any further, we should start with the history of cells. And you can't understand America's future if you don't understand America's past. So we can't understand where we're going with cells and where we are now if we don't understand where we came from. So come along. So there are so many people that can be credited and that should be credited with with what we know of cells today being our early contributors. But we are just going to focus on just a few of them. And as you will see, that many of these guys are actually rivalries, and they observe each other's work and they made things better from each other's work. And so basically they climb on top of each other's shoulder to give rise to themselves. And that's something that's constantly happening in science. The first guy we're going to look at is Robert Hooke. Robert Hooke was actually an English scientist. Perhaps his most famous microscopic observation was his study of thin slices of pork. He came up with a compound microscope, and he observed pork and noted what he saw. And he wrote, actually, and I'm quoting Robert Hooke. This is not me talking. I could exceedingly plainly perceive it to be all perforated and porous, much like a honeycomb, but that the pores of itself were not regular. These pores, or cells, were indeed the first microscopic pores I ever saw, and perhaps that were ever seen, for I had not met with any writer or person that had made any mention of them before this. Hooke had discovered plant cells. More precisely, what Hooke saw were the cells walled in a cork tissue. In fact, it was Hooke who coined the term cells. It's like this box-like cells of cork reminding him of the cells of a monastery. So Hooke also reported seeing similar structure in wood and in other plants. Shortly in 1678, after Lewin Hooke's written to the Royal Society with a report discovering little animals, specifically bacteria and protozoa. Hooke was actually asked by the society to confirm Leeuwenhoek's findings. He was successful, or he successfully did so, thus paving the way for the wide acceptance of Leeuwenhoek's discovery. Hooke noted that Leeuwenhoek's simple microscopes have or give clearer images than his compound scope, but found the simple microscope difficult to use. He called them offensive to his eyes and complained that they were much strained and weakened the sight. All right. Well, clearly, rivals. It's like, well, his microscope works a lot better, but there's still some flaws. So Hooke discovered an Asian guy who actually discovered plant cells, and he named the cell actually coined the word cell. And Anton van Leeuwenhoek was the first guy who actually went after animal cells, but he looked at bacteria and protozoa from looking at pond waters. Well, the next player is going to be Schwann, or Theodore Schwann. He was a zoologist who observed that the tissue of animals had cells. So in 1838, Schwann became familiar with Matthias Schleiden's microscopic research on plants. Schleiden described plant cells and proposed a cell theory, which he was certain was the key to plant anatomy and growth. Pursuing this line of research on animal tissues, Schwann not only verified the existence of cells, but he also traced the development of many adult tissues from early embryo stages. All right, so Schwann, zoologist, Schleiden, botanist, Schwann used Schleiden's work, but in the context of animals, to see if what he saw with plants is also true for animals. One of the sometimes seldomly credited person is also Rudolf Virchow. Virchow was actually a German scientist. He also reported that every living thing is made of cell and made up of a vital unit known as cells. He also predicted that cells come from other cells. So Virchow is credited with several very important discoveries. His widely most known scientific contribution is to the cell theory, which we're going to talk about shortly, which is built on the work of Theodore Schwann. He was one of the first to accept the work of Robert Remarque, who showed the origins of cell was a division of pre-existing cells. So this brings up the whole idea of cell division, something we're going to talk about, in the following unit. So basically, these guys actually they paved the way to what we know about cells, and specifically also not just what we know about cells, but how we know them. Therefore, the whole idea of using microscopes. The cell theory is a name given to three statements about cells in general. But these are put together after looking at the work of many scientists, but Schleiden, Schwann, and Virchow are credited for it. All living things are made of cells. That means going back to our very acronym GROUT chart from the beginning of this school year for the characteristics of life, being made of cell is one of the most important requirements of all living things. Cells are the smallest unit of life, and new cells come from older cells or pre-existing cells. Cells can produce and transport energy within a system. Who we are is basically in our genetic code, which is transferred from one generation to the next in the nucleus of the cell. Alright, so looking closely at a cell, there are some basic biochemistry that are true for all living cells. I mean, I like many other science branches of science, but when it comes to biology, specifically cells and molecular biology or genetics, that's a word that really fascinates me, especially because we have so much diversities in cells, and yet also very close similarities. For example, look at you. All right, you as a person, you are home to about over 100 trillion bacteria cells, which pretty much outnumber your very own cells in about maybe 1 to 10. So that means that our body is actually an ecosystem. So we are host to various forms of life in the forms of little bacteria. Um, it also means that you, or you and I, for some definition of the word you, actually consists of both of the major types of bacteria, prokaryotic and eukaryotic. All cells come in one of these two broad categories, all right? Either you are single cell organism of the domain bacteria and archaea, or classified as prokaryotes. Pro actually means before, and kary actually means nucleus. So when you look at it, this prokaryotes mean, before nucleus. All right. So the question is, are all prokaryotic cells single cell organisms? Well, that depends, and that's a topic that's quite debatable today because there are research in the points in different directions. All right. For your understanding for this unit, prokaryotes, they are very simple cell organisms. They have a membrane, sometimes a cell wall. They have cytoplasm. They have strands of DNA. So again, very simple. A strand of DNA, very simple. They have ribosomes because they need to make proteins, and we'll get to that later. Also, usually bacteria, unicellular bacteria. All right. Prokaryotic cells do not contain a membrane-bound organelles or nucleus. Okay? So basically, everything is just one big spaghetti. All right. The word prokaryote, like I said before, means before nucleus. So prokaryotic cells have no nucleus. They have no membrane-bound organelles. We will see about the other type of cell, a group of cells, also known as eukaryotic cell. All right. So animals like you and myself, I know some of you like to think, I'm not an animal. Well, you are. All right. Animals, plants, fungi, and other protists are also called eukaryotes. EU, which also sounds like YOU, means true, and are made of all eukaryotic cells. All right. So you meaning true, and kary, like we said previously, means nucleus. So true nucleated cells. See that? So eukaryotic cells, they are composed of four main parts. They have cell membrane, the outer boundary of the cell, the cytoplasm, the jelly-like fluid interior part of the cell, and they have nucleus, which is the control center of the cell, which contains the cell DNA, or aka chromosomes, which have organelles or little organs that carry out specific functions and that are membrane bound within that cytoplasm. So this is a picture of an animal cell. As you can tell, there is this part is your plasma membrane, the outer part. You see the outer part. Let me see if I can. There you go. Let me get a better color. So here is our plasma membrane, the outer part, all of this. All right. So when you enter the cell, you will find this is our nucleus, and you have your other organelles like the ER or the endoplasmic reticulum, and you have your ribosomes here, and some of the ribosomes are also studded on your ER, and you have your Golgi apparatus, and you also have your vacuoles, and you also have your mighty mitochondria. So you have all these organelles that are little tiny organs, as is actually literally defined, working together in an animal cell to maintain structure and functions. All right, plant cells, they have some similarities with animal cells. Okay, if you look at this picture, you also have a membrane, which is the blue line. Oh, and I also have a blue line too. The yellow lines you see on the side also. Oops, those yellow lines. Let me guess. I got a yellow. These so somewhat of a yellow. Those are your cell walls. So animal cells do not have cell walls. Plant cells have cell walls. Okay. Also, you will notice still they also have a endoplasmic reticulum. They also have a mitochondria. They have a chloroplast that animal cells do not have. You also have a nucleus. As you can see, the nucleus is not really centrally located in a plant cell because you have this giant vacuole, like this giant water tower that is inside of a plant cell. And that serves a purpose because when you look at it, we don't have water all throughout the year. You know, we have different seasons, or we have water, but we have different seasons, and that brings about shortage of water. Plants use that. They have this giant water tower to just store water for when they need it too. Um, because plants cannot walk from one pond to the other pond to get water like animals would. So let's do some side-by-side comparison. First of all, look over at this picture. This is I need a better ink. Sorry. This is a eukaryote, a lot bigger and a lot more complex looking than a prokaryote, which is much simpler. As you can see, prokaryotes are simple cells, eukaryotes are complex cells. Alright, if you can't tell, look at your body and see how many different systems there are and how hard those systems work together with all that, trillions of cells, and how each trillions of cells of a system can work with the other trillions of cells of the other system to maintain homeostasis. Right? But let's focus on prokaryotes right now. There's no nucleus, they have no membrane-bound organelles, and they are mostly bacteria. Alright, eukaryotes. They have everything that a guy doesn't have: membrane-bound nucleus, membrane-bound organelles, many other forms of life on Earth, like animals, plants, protists, and fungi. Alright? What's true, though, to keep in mind is that all living things are made of cell. All living things are made of cell, and all cells have DNA because that's where the instruction for maintaining that cell comes from. Because cells need energy, and they also need to maintain their own safety and survival. They produce proteins. Also, that's done by the ribosomes. They have a plasma membrane, which regulate what comes in and what leaves that cell, because the cell doesn't just want to do an open door policy. Anybody can just come in and destroy me. No. They have this general workspace or cytoplasm where many things occur. Depending on what cells and what group you fall in, that determines what happens in that cytoplasm. Even a little bit more detail on the classification between prokaryotes and eukaryotes. Let's look at more. Alright, so we said it's unicellular generally. Mostly, the DNA for prokaryotes is usually circular. Organelles, they are not membrane bound. A nucleus, they do not have it because pro mean, before. Carry means nucleus or before nucleus. Typically, it's very small cells, and like we said before, bacteria. Eukaryotic meaning true carry nucleus. True nucleated cells are mostly multicellular. Occasionally, you might find some unicellular, but mostly multicellular. They are organized into chromosomes. All right, and that means you have this looking thing, or sometimes you have, and this is basically double-stranded DNA. You have membrane-bound organelles, and this nucleus, because the DNA is in that nucleus, we know for humans and plants, for example, and they're typically larger cells than prokaryotes, and plants, animals, and protists and fungi actually occupy that area or category of cells. So in short, we just talked about three types of cells, basically, even in that just short within the last 10 minutes. Talked about bacteria cells, which are prokaryotic, plant cells, which are eukaryotic, and animal cells, which are also eukaryotic. We will look at, ignore this, it's still unit 3, sorry guys. We'll now look at the different classification levels of organization. Sorry. Well, we can know a lot about plants and animals from studying their cells. Many of the world's deadliest diseases are pursued and studied by actually looking at them at the cellular level. So that to answer the question, why do we study cells? Well, because cells actually form tissues, and tissues form organs, which give rise to bodies, which are pretty much made up of cells. Alright? Alright? And then all those cells actually give rise to the organisms. And the cells do all the work of life to maintain, the functionality of that overall organism. So this slide is just to give you perspective on the size of cells. Many cells are so small that you cannot even see them without the use of a microscope. I mean, look at, you're looking at the diameter of a human hair. That's larger than the average cell. So if you really wanted to study anything about them, that's why the study of cells always are involved with the use of microscopes. So when we look under a microscope, the cells that we look at fall under one of these two categories: a unicellular or multicellular. It's a unicellular, meaning it's made up of a single cell that's capable of performing all of the characteristics of life, regardless of its size or its simplicity. There are many unicellular organisms that are dominant on life, or dominant on Earth. So these cells are still able to grow, respond to external stimuli. They're able to respire to create energy. They're able to reproduce themselves. They're able to form on an organizational level, use energy. They're essentially made of cells, and they adapt or evolve. Okay, so as simple as they might be, they're also able to carry out all those characteristics that we described earlier in the school year. It's a multicellular, meaning it's made up of many different cells that work together to maintain the function of that organism, carrying out all those characteristics. They're usually made of cells, so because they're multicellular, they have a much more complex organizational level. The simplest part of that is cells, and a group of cells make a tissue, a group of tissue make an organ, a group of organs makes organ system, and that forms an organism. So, for example, you can have a muscle cell, and a muscle cell can put muscle tissue in an organ. For example, like the heart, which is made of mostly smooth muscle tissues, and you have an organ system, which is a cardiovascular system, which also involves the heart, and also cardiovascular system work with other organ systems to maintain the beating of your heart and the exchange of gases and functioning of your body, and so you can run, eat, talk, and do all the things that you do. Cells in these organisms develop in different ways to perform the specialized functions. For example, white blood cells, muscle cells, neurons, all of these act in different ways. They divide differently. Some divide more frequently than others. Alright, this is just an example of pictures under the microscope. Is an onion cells. We have the privilege of actually looking at this. And one thing I'm actually going to have us try to do this year is for you guys to actually design a lab that you actually can stain and look at these cells under the microscope. Right? For example, we will have to sample certain parts of our classroom and see what kind of cells we can pick up and see if we can identify those cells by being able to stain them with RAM stain. And that's just something that's coming ahead of us. Alright. The cells of an organism must accomplish 3 tasks. They must actually be able to make energy. Alright. They must be able to actually make proteins and make new copies of themselves. In actuality, it is actually the little guys, also called the organelles, that are actually doing all of this work. But basically, the organelles must work together to maintain the overall state of the cell in order for these 3 important jobs to actually be accomplished. So cells have 3 jobs: make energy, all right, make proteins, and make more cells. We're gonna look at now some properties of animal cells and plant cells, and see that there are some things that overlap and some things that are quite specific. So welcome to the city of cells. Basically, this is what type of cell? Well, if you guess animal cell, or if you say animal cell, that's correct, because this is an animal cell. And as you can see, there's so many vital parts. There's so many things happening in here that makes the working of the cell together just something that seems so effortless, but is so intricate that as complex as we are able to do things beyond our understanding because of what these little guys are doing. So all these little things you see going around, moving around, colorful dots here and there, they all serve a very vital function. Just so I want to introduce something to you that you should be thinking about right now. In a few days, you will be actually asked to start a project, and a project is called the Cell City Project. Um, we're gonna I haven't given you the platform yet that you're gonna use, or the medium that you're gonna use, or what type of media choice you're gonna use to create this. But essentially what it comes down to is that you will be creating a you know project that will look at different aspects of the cell. All right. You will compare the functionality of each organelle with something that has a general theme. For example, like an amusement park, a car, your home, your school, a specific city, company, agency, office, you know. So you will have to do that, and I will give you an example all through this lesson how to develop those, so to help you out. So that's something you want to start thinking about for those of you that like to work ahead of your teacher or so. So what is inside of a cell? Well, when we look inside a cell at a closer look, or when we take a closer look inside of a cell, all right, we see that all cells have a plasma or plasma membrane, or also known as cell membrane. And beyond the cell membrane is a cytoplasm, which consists of what we call the cytosol. Okay, and that cytosol is the aqueous component of the cytoplasm of the cell, within which you have your organelles and particles that are suspended. Aqueous solution, all that is intracellular fluids. All right, this is very important for your organelles because it keeps them sort of stationed and not colliding to each other back and forth. All right, so it's like a plasmic liquid, but it's not liquid, so that things will just float about. Okay. Each organelle performs a very specific job to help the cell survive. All right. All right. So now let's start looking at each individual organelles for the most part. So the first organelle we're going to look at is a nucleus. All right. The nucleus is the brain, quote unquote, of the cell, because it contains your DNA, and the DNA contains the instruction for making proteins and giving instruction for any other vital things that need to be created for that cell. The nucleus consists of a nuclear envelope, which when you look in this picture, this area is your nucleus. But when you look around it, you see these dotted area. All right, that's your nuclear envelope, which is also, this, when you look at this section of drawing. All right, so you need a nuclear envelope, and the nuclear envelope serves as much more of a further protective barrier. So in case anything made it past its cell membrane, it doesn't just automatically get to the main blueprint of the facility. So as to not being able to easily just overcome this cell. But when you look further into into the nucleus, you also see this little part right here. That is called the nucleolus. All right, it is in the center of the nucleus where the actual master instruction for proteins are generated. So an analogy of the nucleus is for prison is basically since we are speaking of cells, is the warden's office. And that's where all the details of what happened at that prison or what is going to be done at that prison will originate. All right. And the ribosomes. All right. There are two types of ribosomes. All right. The free and attached. So when you look into the cell, you have these free float. No, those are not ribosomes. Sorry. You have your free floating ribosomes. These ones. There you go. All right. You have your free floating ribosomes, and you have your attached ribosomes, which are studded. If you look into the zoom out picture, studded along the endoplasmic reticulum, which we will talk about soon. All right. The ribosomes are where proteins are assembled. So it's basically the work assembly bench of making proteins. The cell's main job, really, well, it has three jobs. One of its main jobs is making proteins, like I said earlier. So that's where that happens. The instruction comes from the nucleus, and it goes to the ribosomes, and the ribosomes are the chefs, and they make up whatever the chef, what the chief chef recommends should be made. All right. So going, back to our prison analogy, this is the kitchen or the cafeteria staff making food for the inmates. So the warden said, "Hey, this is what what's being fed to these guys this month, and each day that month, the chefs will make that. The ribosomes will make whatever the DNA says is protein that should be made." The endoplasmic reticulum, also shortened as ER, all right, is made up of many layers of folded membranes. As you can see, these little intricate folds that you see. Basically, this is divided into two parts. There's the smooth ER and the rough ER. Smooth ER, as you can see, if you look at this section, all right, it's smooth because it does not have any of these ribosomes attached to it. It has no ribosome attached to it. So the job of the smooth ER is to make lipids for the cell membrane. Lipids, we just remember we did what, 4 basic macromolecules or biomolecules, and lipid was one of them, also known as fat, and we said it's also part of your cell membrane. All right, so the lipids made for the cell membrane is actually made in a smooth ER. The smooth ER is actually used for detoxifying drugs or breaking down the toxin out of drugs that people, for example, if someone is overdosed on a drug, and the lipids actually, the smooth ER is actually, tasked with being sure that the cells doesn't die because of the toxin from that drug. All right, the rough ER is rough because it has these studded ribosomes attached to it all over. It's like an ER with acne, so basically. The rough ER has the main job of the rough ER is to pretty much modify the proteins that are made from the attached ribosomes. You see why? So ribosome makes the protein. The rough ER says, okay, let me make sure that you made them correctly and make sure they have the right shape, right size, the right folding, and all that good stuff, so they can serve the right function. So this would be the different corridors or hallways in the facility or prison that connects all the different parts, which also have spaces for making sure that things are happening. You have the CO office, which oversees making sure everything's happening correctly. So all of these are connected. The Golgi apparatus, also known as the Golgi body. So it's not Golgi, it's Golgi. Okay, the Golgi apparatus or Golgi body is made up of stacked membranes. They pretty much customize the proteins as they needed to be performed to a specific job. So meaning that the protein comes in, and basically if there was an immune cell protein, the Golgi apparatus is going to put that stamp, and so then the cell knows where to ship that to ship to which part of the cell and outside the cell. So basically, if it was proteins to use for brain cells, it would stamp it. So the final modification before that protein actually leaves the cell is done by the Golgi apparatus. So the Golgi apparatus also act like the post office of the cell. It sorts, package, and ship things or proteins wherever they need to go. I mean, it is the one who actually customize them for a specific place and destination. So why not just sort them out in where you know, customize them and pack them and ship them out. All right. So this is example will be the mail room at a prison where you have good inmates and also model inmates and employees working together to sort, package, and ship things that are going out of the prison. Your lysosomes sounds like Lysol, right? Lyso, right? Now, what do we use Lysol for? We use Lysol to clean things. All right, lysosomes clean up these cells. They are filled with enzymes. Enzymes we also say are proteins. All right, so these chemicals are used to destroy all debris that are found in the cell. So the useless part of in the cell, it eats up all the garbage, all the gunks, and then ship them out of the cell. Basically, it's the trash disposal system of the cell. So this is like the custodial staff at a prison or prisoners who are responsible for the general cleaning and maintenance of the facility. They are equipped with cleaning supplies like Lysol, and just like the cell have enzyme or the lysosome have enzyme to break apart deadly agents or deadly disease-causing things and things useless to the cell and ship it out of the cell. All right. We also now have the vacuoles. All right, the vacuoles sounds like vacuum, right? Ha! I almost not be able to spell vacuum. All right, so but it's not a vacuum. But think of a vacuum. Vacuum is an empty space. But a vacuole is also a space, but it's not empty. It has stored waste or excess material that the cell does not use, such as water. All right. In plants, there's a larger vacuole than in animals because this is an animal vacuole, that's a plant vacuole. The central vacuole in plant is one of the most distinguishing part of a plant cell from an animal cells. All right. In animals, there are smaller ones of these because it doesn't have a large one. And I think this serves an evolutionary trait or reason or survival reason, sorry, is that plants do not move around to search for water. So when it's harder for the roots to reach the water sources, they draw from their vacuoles, and that's some of the reason why you lose leaves on plants during the autumn time and fall time before the winter starts. Animals, if I'm thirsty, I'll find me a water source, and most animals will be able to find themselves to a water source to get replenished. Mitochondria. All right, the mitochondria is mitochondria. I like to call this guy the mighty mitochondria because it's the powerhouse of the cell. All right. Basically, they have two membranes, the inner and outer membranes. All right, and actually, you should check out many of these organelles. I have added some 3D images to your nearby presentation. You can actually swivel and zoom in and look at these. What do I mean by inner and outer membrane? Basically, the job of the mitochondria is to provide energy by converting chemicals from food into energy that the cell can use, also known as ATP. All right. So basically, you eat food. All right. It takes your nutrient out of your food and break it down further and creates ATP. At one point, it was actually thought that the mitochondria was a free living organism since it has its own DNA. Yes, this is an outsider. This guy has his own DNA and it replicates independently from the cell. Alright? Chloroplasts. Every time I look at these guys, I think of pancakes and I get hungry. Oh, sorry, I did not give you I first did not give you an analogy for the mitochondria. In the last slide, the mitochondria would be the power plant that supplies a prison with electricity. Okay, so now let's come back to the chloroplast. This is only found in plants. Okay, excuse me, algae and other cells. All right, they convert sunlight into food to be sent to the mitochondria. Well, look at it this way. This is one of the main places excuse me, you have photosynthesis occurring. So it takes the sunlight, plants absorb it, it goes into the chloroplast, and they transform that into what? If you said glucose, that's correct. All right. So we eat that glucose. All right, and that glucose goes to the mitochondria. That's what this is saying. There are two membranes, the outer and thylakoid membranes. Okay. The chloroplast also contains your chlorophylls, which give the pigments or the grain coloration that we see in plants, or the yeah, sorry. The chlorophyll gives that grain coloration that we see in plants, and it also contains its own DNA. Also, talk about outsider again. The chloroplast pretty much is found only in plants, and it is a key player in photosynthesis. It acts like a solar panel, if you want to say, of a prison, just in case the regular power goes out. These solar panels have taken the sunlight and turned them into electrical energy that can then be used as a backup source of power for prison. If you notice, I'm keeping one thing here, prison. I'm not just using all these analogies of different things that I can think about. Just keep it focused on a specific theme. All right, the cytoskeleton. All right, first of all, let's break something up. I should have done this a long time ago. Cyto means cell, by the way. If ever you hear the prefix cyto, it means something to do with cell. So cytoskeleton, that has to do with the skeletal system of the cell. All right, and what do we know about skeletal system? It supports general structure and overall standing of an organism, or of a being or entity. So the cytoskeleton supports the cell. It is made of tiny little tubes called the microtubules. Micro means tiny, and tubule, tubes. And these tiny threads, also called micro, means tiny, filaments. Sounds like how you call it? I forgot the word. But microfilaments, they act to keep the cell in a 3D shape. They also aid the cell or help the cell in movement, because again, our skeletal system helps us to move. So does the cytoskeletal system of the cells. Okay, as you can tell from these pictures, these are all parts of the cytoskeleton of the cell. These act like the rods and steels that we do not see in a prison wall, but they keep up the structure of the overall prison. All right, so you don't see many of these, rebars that are put into buildings, but they are there because they keep up the general overall structure of that building. And if you look at skyscrapers, for example, they use a lot of those, and skyscrapers actually do move. They sway back and forth, because if they didn't do so, they would just collapse. And so you can look at that as an analogy as well. Sorry. Centrioles. Centrioles actually come into play into animal cells reproduction, or making sure the main job is making sure that the chromosomes are organized or lined up so they replicate correctly. They act like the foreman at a prison, making sure that every maintenance job is done correctly, making sure that there's no compromise with the integrity of any part of that structure, so you don't have to get an easy breakout. All right, so centrioles is actually involved into cellular reproduction. They're found near the nucleus, and they are not implanted, just only in animals. All right, cell membrane or plasma membrane. All right, these guys surround all your cells. They're found in all cells. They regulate what enters or leaves the cells. They provide protection and support, and they're made of lipid bilayer. All right, so when you look at your cell membrane, all right, it has phosphate head and lipid tail, phosphate head, lipid tail, phosphate head, lipid tail, and it's bilayer because you have two layers. All right, and because this inside part is lipid, and lipid is fat, and fat is insoluble with water, therefore it's called hydrophobic, and we already learned that term in last unit. The phosphate head are actually hydrophilic. That means they can actually combine with water. If you think about it, the inside of a cell has water, the outside of the cell has water, but you don't want water in the middle part of the cell. All right. So looking at the analogy for this is like the guard, all right, at the gate, making sure that not everyone can just enter or leave the prison, just the people who are allowed. Cell wall. This is only in plants. Okay, it protects some cells, plants, algae, fungi, and prokaryotes. Outside cell is found outside the cell membrane. So the cell membrane is actually inside of the cell wall of plant cells or those cells that have cell wall. They allow water, oxygen, carbon dioxide, and some other substances to easily go through. The main function is to provide support and protection for all cells. So this is the fencing around the entire prison. All right, and the fencing is actually outside of the guard gate, so they can make sure that nobody's just get through. Even if you do, the guards is, they're there watching. And also if you were to get past the guard, theoretically the fence should stop you, because the fencing have electrical barbed wires or razor wires that should stop you, and electrify that should also shock you. All right, I'm not trying to be mundane and morbid here, people. So how are we going to study all of these cells and everything about cells? Well, part of this unit you will learn how to use a microscope, and to use a microscope you got to know the different parts of a microscope. So you just don't, because we just can't look at cells. So number one, this part of the microscope is called the body tube. All right, and my writing is gonna be horrible. That's the body tube, and the body tube is connected to what we have called the revolving. I'm just gonna review that revolving nose piece. Doesn't it look like a nose? It might. These three guys, these are called objective. All right, these are your objective lenses, and they go in power of magnitude. This is the highest, and this is the lowest, and this is the median. All right, and number 5 is, sorry, that's not what it is. Number 6 is your stage clips. Your stage clips actually hold down whatever specimen you have on a slide. And number 7 is your diaphragm of the microscope. All right, sorry, this is a word that always gets me. That's a P, diaphragm. So the diaphragm regulates how much, the amount of light that's also coming through. Number 8, this is your light source. All right, so if this is plugged in, the light bulb is located under here, that then shines through the diaphragm and out through to your specimen. Number 9 is your ocular lens. All right, ocular have to do with the eye, ocular lens. And number 10 is the arm. Yeah, technical term. You hold the arm to carry the scope. All right. Number 11 is the stage, where everything unfolds. This is where the drama takes place. Number 12 is your course adjustment knob. All right, so this is the first knob you turn to keep things in focus on the big picture. And then number 13 is your fine adjustment knob. And this actually, my friends, is when you actually focus on a detail. Number 14 is the base of the microscope. So to carry the microscope safely, you have to hold it by the arm and the base at the same time to support it to carry it. All right. So for the survival of the cell to be certain, there is a tight regulation of what enters or leaves that cell. All right. There are various parts that make sure that this happens. And does that remind you of a specific organelle that we just talked about? If your guess is sorry, get rich, the plasma membrane or the cell membrane, that is fantastic. All right. I would recommend you download. It's called, I think, self defense or defense. So I think, yeah, it's called self defense. I would recommend you download this app. This is an activity we're gonna do in class, and so if you download this app on your own and you start, it's a game really. You build a cell membrane and it guides you through several different tasks, and that shows you how things move in and out of the cells and which components are used and which components are necessary. So I would say if you want to get a head start practice on this activity, please do so, so that way when we do it in class, it can run a little smoother, or smooth. It can run a lot more smoothly, so we don't have to, sit around and work out those small bugs. So let's focus on the cell membrane for a second here. When we talk about transport, because why do we pick the cell membrane for all the other organelles? We're talking about transport because the cell membrane actually is the gateway by which things enter and lead to cell. This is the border, right? So the border of a country, you got to go through custom agents to get into that country. So welcome to the cell border, okay? And this system keeps a tight border, all right? So the cell membrane, you have to pay close attention to all the details because this is a big part of your unit. Cell membrane actually contains lipids and other molecules. All right, they're up. When you look down in this picture, I already described to you earlier. This phosphate head, lipid tail, phosphate head, lipid tail. The cell membrane is described as a phospholipids, phosphate head. All right, so phospholipids. All right, bi-layer. Phosphate head, okay, this part right with the yellow. Lipid tail, this part underneath it, and it's a bi because it's two layers, layer 1 and layer 2. See that? All right. There are protein embedded in the membrane. They form channels and pumps to move material through. And you're like, okay. So here's a protein channel. You also have things can come in and things can, things can go out and things can come in. So when you look at a cell, this is the inside and that's the outside. All right. So on the outside, we also have carbohydrates that are attached to these proteins, and the carbohydrates are pretty much like the sensor or sensory system of the cell membrane. Basically, they feel around and see if there are other cells nearby, and you can recognize each other that way. So imagine closing your eye in a dark room, large room with many people, and you don't want to bump into each other, and you're playing, feel in the dark, if that's a game. The cell membrane is also called a mosaic because it has so many different small parts that make the whole. As you see, you have the phosphate head, lipid tail, protein channel, protein pumps, carbohydrates, and all of these work together to make this one unit function well. All right, things moving in and out of the cell. All right, the cell membrane. So we're still talking about the cell membrane. So the cell membrane is called selectively permeable, or it has a function called selective permeability. So selective meaning picky, okay? So I'm a picky eater, so I'm a selective eater. Permeability. So far we have the same letters as in permeate. All right. So permeability means selectively permeate, or pick and chooses what is permitted in and out of the cell. So the cell membrane gets to pick and choose which molecules can pass through, and that actually is dependent on the size of the molecule, what charge or what type of ion is it, positive or negative charge, and solubility meaning how much is inside the cell and how much is outside the cell. If too much is in, we wouldn't allow this. The cell likes to have an even balance of things in and out of the cell. All right. So substances that can move in through or that can move through are non-polar molecule, fatty acid, vitamins A, D, and K, steroids, oxygen, carbon dioxide, and water. Substances that cannot really move through are sodium, potassium, calcium, hydrogen, ion. The polar molecule like polysaccharides, polyamine, many. This is a complex carbohydrate. Big molecules. Amino acids, the building block of proteins, glucose, nucleic acid, and those sort of things. They cannot these guys cannot easily move in. That doesn't mean the cell doesn't take them in, but they cannot just easily move in. All right, the cell wall. All right, the cell wall is made up of carbohydrates and proteins. It is found only in plants and it's made of cellulose, O's, as you can see here, carbohydrates. Okay, the cell wall make up the carbohydrates and protein, and then make the carbohydrates and protein, and then send them to the surface where they can be assembled into the cell. So basically, this is one of the structures that gave the rigidity of plant cells and the structure of a plant in overall. The cell wall sort of acts like the cell membrane, but not exactly. They don't have exactly the same makeup, but it still regulates what comes in and what leaves. So the cell wall actually allows water to come in, water to leave. It also allows sugar molecule to come in and to leave, oxygen to come in to leave, carbon dioxide to come in to leave. So those things are easily movable through the cell. Well, here comes our H word from Grouchard, homeostasis. So here comes this H word again, homeostasis. I told you guys earlier on during the school year that looking at all these characteristics, they will play roles in different aspects of our school year. All right, so homeostasis, again, just to remind you, is maintaining a stable internal condition. Cell must maintain suitable internal condition despite of the changes happening outside. So even if it is cold, the cell cannot, the body cannot allow, the cells inside to freeze to death. So something happens. So when it's too cold outside, you start to shiver, so your body can warm up. If it's too hot outside, you start to sweat, so you can cool off. All right, this is accomplished by cellular transport, or the movement of molecules into and out of the cell to meet those desired needs. All right. So the transport is actually regulated by the concentration of intracellular fluids in between cell fluid and extracellular fluid, or outside cell fluids. All right, when we talk about concentration, we're looking at mass of the solute over the volume of the solvent. So you should be able to actually calculate that, because if you have 15 grams of salt into 20 milliliter of water, what is the concentration? You should be able to solve that. The solvent in this case is typically water. We say water is a universal solvent because it dissolves many things. So when you look, this is inside of the cell and the outside of the cell. The goal is, things can pass through the semi-permeable membrane to make sure there's a balance there's a balance on what's on the inside and what's on the outside. So positive and negative feedback. This system works very well using these feedback system. Homeostasis is maintained, or regulation is maintained using either one of these two. So the positive feedback will intensify the condition. So positive, that means something good, all right? But not as we think of it as good. So for example, drug addiction. The more drugs you take, it intensifies the feeling you get. But that doesn't necessarily mean more drugs is good for you. So the more craving means more drugs needed. So that craving will not be satisfied without that drug or something that acts like it. You understand? So negative feedback is the complete opposite of that. It acts to counterbalance or stabilize the internal conditions. So in your house, all right, if you put a thermometer or thermostat to 70 degrees. Once the house temperature falls below 70 degrees, the heat source will kick in. If it goes too high, it gets to 80 degrees or above 70 degrees, the cooling, either the heat source shuts off or it turns on the cooling source, depending on what season you are in. So same thing happens in a cell. So too much salt moves into the cell, and then the cell will respond by increasing the water content of the cell to decrease the salt solution, to dilute the salt. So most of what happens in the cell is controlled by negative feedback. Negative feedback loops, not loops, by the way, loops. All right. So this is common with I always make a statement about black people and or African American people, for those politically correct people out there, and high blood pressure. So, and that happens with anybody too, but just that most African American diets are actually rich in salt. So the more salt you take in, that's increased salt concentration in your blood vessel, and then the system then flush that system with more water, and because you have an increased water volume, that increases the blood volume, and therefore the pressure increases, and so the heart is overworking to actually pump that stuff out. So this is more salt, more water to dilute salt. All right. So negative feedback. So now let's talk about diffusion. When something is diffusing, it's trying to fill an empty space where it's coming from an area of larger gradient to an area of lower gradient. Meaning it's going from a high concentration to a lower concentration. It uses no energy to actually do that. It does not require any energy to do that. The way, for example, if I stood in one corner of this room and I spray an air freshener in the air, pretty soon, within seconds or minutes, you will start to smell the fragrance in the opposite corner, because the particles or the molecules of the air freshener will try to fill all the open spaces that it can get to. So it's diffusing through by going through this, the membrane or this cross section. So the goal is to move and fill this empty space, or most like balance out. Once you reach an equilibrium or balance, like in this picture, things will still keep moving back and forth. But if one moves out, one moves in, one moves out, one moves in. So there's a net balance. So this is at equilibrium. So what's moving in, one's coming out, one's moving in, one's coming out. Requires no energy. This is called passive transport. So passive transport is moving from an area of higher concentration to an area of lower concentration without needing to use any energy at all. All right. There are different types of passive transport. All right. Just to illustrate a point of passive transport to you. Or passive, you're not really using much of any energy. You're not even using any energy. Intentionally. So see yourself at the top of a hill on a bicycle or a skateboard. You don't really need to propel yourself. All you need to do is sit or stand and let gravity takes over. All right. So osmosis is a type of passive transport, and osmosis is only concerned with water. All right. It's a diffusion of water. It's a type of diffusion that's only done with water. And basically, water has this thing where if it is in an area of high concentration, it will always try to go to an area of lower concentration. So if there's more water in the cell, it will start to pump water out of the cell. If there's more water outside of the cell than in the cell, it will try to pump water inside of the cell. All right. This requires no energy whatsoever. Facilitated diffusion. This is when you actually use proteins to facilitate diffusion for specific molecules that cannot easily cross the cell membrane by creating channels. Remember those protein channels that I show you in the cell membrane? Yes, that's facilitated diffusion. It still doesn't. Protein channel is like a tunnel. So the perfect example of this is going into Manhattan. You will not go into Manhattan unless you go through the tunnel. system. So, the tunnel system just allows things to come in and out. All right. It's not allowing. It doesn't really require the use of inner energy to do this. So molecules move from where molecules are high in concentration and they move to an area of lower concentration. So it's just that movement back and forth to make sure that they reach an equilibrium. But again, osmosis only with water from high to low concentration. Facilitated diffusion uses protein channels to move things from high concentration to lower concentration using no energy. Therefore, it's called passive transport. Passive transports do not use energy. All right, coming back to osmosis, there's this thing called osmotic pressure. Okay, because cells always try to keep a balance or try to maintain equilibrium of what's in and what's out of the cell. So when you look at our red dots, all right, our sugar molecules and our blue dots are water molecules. So when you look at the system, there's more sugar over here than over here. So the goal is that, what can go in and out? Well, water can easily go in and out because this is osmosis. So the water would then try to move from here to create a balance of water over here to water over here. All right. You notice the sugar molecule are not moving because osmosis has to do with just water, not sugar. Too much water in a cell can actually cause the cell to burst, which we call lysis. All right. Because too much, think about the cell as a balloon. You add too much air into it, it will burst. You add too much water, even in a balloon, it will burst. Too much out, it will shrivel or crenate. All right. Too much water out of the cell will crenate or shrivel. It's like when you're dehydrated, you start to lose water, so your cells start to starve. Take the air out of a balloon, it shrivels up or crenate. All right. So the movement of water, the movement of water in and out of the cell is actually regulated also in a way that water is readily able to move across the membrane through protein line channels or protein channels. And if the total of concentration of the dissolved molecules or particles inside of the cell is not equal on both sides, there will be a net movement of water in and out of the cell. Whether there is a net movement in and or out of the cell, that also in whichever direction, it depends on whether the cell's environment is isotonic, hypotonic, or hypertonic. All right. So let's look at isotonic. When two environments are isotonic, that means the total concentration of dissolved substance on the inside or outside are the same. All right. So when cells are in an isotonic solution, basically the movement of water in and out of the cell is exactly the same by measure. Okay. So you have equal amount of water and solid that will be moving in and out of the cell at the same rate. So this, for example, in animal cells, they have about close to 1% of salt or saline concentration. So when we have to expose animal's tissue to a solution, we try very hard to make sure that it is about that same concentration. Because if you don't do so, basically what happened in consequence is to damage the cell by actually increasing the osmotic effect on that cell. That means how much water is going to move into that cell. So let's talk about now. Move on to hypo and hypertonic. Hypo means under, tonos means stretching. So in a hypotonic solution, the total concentration of all the dissolved substances or particle is less than the other solution or less than that of the cell. So in that case, what's going to happen is then if the concentration of dissolved solely or less outside of the cell than inside of the cell, the concentration of water outside is going to correspond greatly. Basically, it's going to be more water pushed outside and when a cell is exposed to too much hypertonic condition, there's a net movement of water in the cells. So the cells without walls will swell and then will burst or lyse. So this is very common for animal cells because we don't have cell walls. So excess water is not removed from the cell. If excess water is not removed from the cell, it will burst and lyse. So the cells with walls often then benefit from the pressure that develop in the hypertonic solution. In a hypertonic environment, okay, hyper means over, and tonic again, stretching. So in a hypertonic solution, the total concentration of all the dissolved particles is greater than that of the other solution or greater than the concentration in the cell. So if you have a greater concentration of dissolved solutes outside the cell, this concentration of water outside is going to be then lower because you got more stuff, so less water is more stuff occupied. So as a result, water then will then move inside of as a result, water inside the cell will then float outside to maintain an equilibrium, causing the cell to shrink. So all that stuff outside now is drawing all the water out to it, and therefore the cell now start to shrivel up. So as the cell loses water, they will not have the ability to function or to divide or to move. So hypertonic environments such as concentrated like syrups have been used since the old time of humanity as a preservation for food, because microbial cells would not otherwise be able to actually come in and wreak havoc. Okay, so quick review of hyper and hypertonic. Cells will move whichever is easier and require least amount of energy. Typically that means that water will move to reach equilibrium. So water will always move from hypertonic to hypertonic because there are more water molecules in a weaker solution. All right, hypo, and iso, they are referring to solution. So solute dissolved in a solvent. I know it's a lot to wrap your mind around, but play this back. Replay this if you need to give you a better understanding. All right. So far we've talked a lot about passive transport. Now let's talk about active transport. Active, when you're active, you're using energy. I cannot be active. I'm running up and down. Like yesterday, actually, when I walked about 13 miles yesterday. First time I went to Route 9 and I jogged about 10 miles. 5 miles in, 5 miles out. I came home and I was so starving, and I ate a bunch of stuff. Then I had to take the dogs for about 3 miles last night. And so here's the thing about it, is that all that moving around requires me having energy. I cannot be that active if I don't have a replenishment of energy. So active transport actually needs energy, all right, in order to happen. So it's the movement of particles or molecules from across a membrane, as always, but it's coming from an area of lower concentration to an area of higher concentration. It's moving from a lower concentration to a higher concentration to maintain equilibrium. In other ways, if you're going to move from low to high, you're going to need a lot of energy for that to happen. All right, and this is where our protein pumps come in the transport. So these transport protein are acting as pumps. All right, look at it this way. If you're going to pump water, so this is a water tower down here, and a water reservoir, and there's a tower up here. If you're going to pump water from this tower up here, you're going to need some power source to have that water go up. All right, so when you're going from a low concentration to a higher concentration, it requires a use of energy. And that is active transport in a nutshell. There are two main types of active transport, and there are other subcategories that fall in those, but we're looking at endocytosis and exocytosis. We can break these up. Don't panic. Endo, it has to do with what? Or inside, All right. Let me use my hand. There you go. All right, inside, and exo like exit, so outside. So basically, this side has to do with moving things inside the cell, and this has to do with moving things outside the cell. Let's focus on endocytosis for a moment here. Okay, it's used to bring materials in the cell, like we just said. The cell membrane folds and form these little pockets, and the large molecules are then found within that pocket. Once inside the cell membrane will then dissolve, releasing those molecules, and this requires energy. And this includes pinocytosis and phagocytosis. All right, well, let me draw you a picture. So this is your cell. All right, you have this thing here. All right, here's your molecule in there. It needs to come in here. So basically, what is going to keep doing it is going to keep moving in, and then it dissolves through the cell membrane. And when it comes in, so basically, it comes in and it becomes part of the cell membrane, and this opens up, releasing, that material out. All right, so pinocytosis clearly has to do with liquids and coming into the cell, and phagocytosis has to do with solid coming into the cell. So pinocytosis is actually often referred to as the cell drinking, whereas phagocytosis is referred to as a cell eating. All right. Exocytosis is the opposite. Things are moving outside of the cell. All right, so same process, but now this time the thing starts out on the inside of the cell and now moves inches towards the cell membrane, fuses into the cell membrane, and then opens up and releases out whatever needs to come out. All right, and this requires the use of energy as well.